Okay, this video is just to give you a little bit of background on some of the techniques used in the Amoeba paper. This is just an image, their last figure, figure 8, which is kind of what they think is happening with the amoebas. So I just want to make sure you understand the difference between trogocytosis and phagocytosis. So trogocytosis is this idea of a nibble, meaning that the amoeba is biting off or removing a little bit of the membrane, cytoplasm, cell contents from the, in this case, human cell that they are attacking, but they're not engulfing the whole thing. So it's different than phagocytosis, which I'll show you in a minute. A couple important things to remember is it requires direct contact, so attachment to the human cell. And what's shown in red here, these little sticks, is it requires actin. So on the home page, I've linked you to a couple videos, and the first one is showing an amoeba phagocytosing or eating a paramecium. And you'll see that the membranes go out and around, and you can kind of see membrane movement here, and that's actin dependent. So that's part of their data showing that in fact this is a eating function, and it's requiring actin to actually what do I want to say, move the amoeba membrane to the human cell and kind of biting off a chunk. The idea is that then some of these host proteins, or sorry, not host, human proteins will become part of the amoeba cell membrane. And this isn't the best picture to show it, but if we made some red lines here, oh, let's see, let me clean this up a bit. So if we had our human cell with some red proteins, let's say, and the amoeba takes some of those in, the idea is that now some of those red proteins from the human cell are expressed or shown or presented on the cell membrane of the amoeba. And the big idea is that's going to protect the amoeba from the host immune system. Okay, phagocytosis is down here. So this is the picture we just saw. Phagocytosis is this engulfment of some kind of foreign object. I should say external object. So it can be a cell, it can be something else. Phagocytosis is the way we talk about eating. So this again is actin dependent, and what's happening is the whole cell or part of the cell is engulfed by the amoeba, and then it's usually fused to a lysosome for digestion. So a big part of this paper is them differentiating between trogocytosis and phagocytosis. Towards the end of the paper, they use a ROM1 mutant, and the ROM1 mutant is defective, my understanding, in attachment and ingestion. So it's a protease, regulates the submembrane distribution and size of the gal-galnac lectin subunits, and that's shown right here. That's important for attaching to the host cell that it's going to either eat or nibble. My understanding in the paper is that they say this mutant is defective in phagocytosis, but not trogocytosis. And honestly, I don't understand why. I'll reread the paper some more and see if I can figure it out, but that might be something to think about. Also note this is a knockdown mutant, so not necessarily 100% loss of expression of this lectin. They're not mutating the gene, they're just causing a decrease in expression. Cytochelsin D is an inhibitor of actin polymerization, so your actin fibers can't grow and elongate, and so we've already talked about that actin is important for both trogocytosis and phagocytosis. Again, actin is shown as these little dotted lines, so they're analyzing if actin is indeed important, and I think what they're trying to do is really say that you have to have this taking in of part of the membrane. It's not just contact, right? It's not just we're going to touch each other and I'm going to get something transferred. And they do use a lot of labeling reagents. So biotin is just a very common labeling reagent. It has a high affinity to avidin, so you get this really strong bond, and this allows you to stain or detect certain molecules. So it's a way to label the cells. When you see DMSO, this is a control. This is like your negative control. So DMSO is just the reagent that is used to dissolve the other dyes. So it's like PBS. It's just kind of a buffer thing. So whenever you see DMSO, that's your control. Host and DAPI staining are used to stain DNA or nuclei. Though really the difference is host is used on live cells and DAPI you have to fix the cells, but they both stain the nuclei blue so that you can have an orientation to cells. DID staining is a red stain, and the only thing I can glean from this is it's really good because it labels cells very evenly, so perhaps that's why they used it. I honestly don't understand why they used all these different stains. Live dead violet shows you dead cells as purple. And I love this, zombie dyes. When it's a live cell, it excludes the dye. When the cell is dead, the membrane is more permeable, and so the dye can go in. And then you can use flow cytometry and FACS cell sorting, which I'll talk about in just a minute, to distinguish the live cells from the dead cells. So there's markers that they can tell which cells are what percent of cells in a mixture are live versus dead. They use a lot of confocal microscopy, which is just really high resolution microscopy. You can get 3D images, but it's a way to get really nice pictures. And immunofluorescence, immuno always means antibodies that are fluorescently tagged somehow. So again, this is just to show specific proteins, parts of a cell. So you would have your protein that you're interested in, or your part of your cell, and you would use a set of antibodies, the immuno, with fluorescence to light it up. And the nice thing is you can have multiple colors at one time. So here's all your blue DAPI nuclei, and then they also have a green and a red fluorescent marker so that you can see different components of a cell. Or in this case, different cells are labeled different colors. The amoeba is usually green and the human cell is red, so that you can see this movement of proteins from the human cell to the amoeba. Flow cytometry is just a way to sort or discriminate between different cell populations from a mixture. So you can have things labeled with different markers, and the flow cytometry machine will read these, and so it can give you kind of counts or percentages of cell types, and it can actually sort different cell types into different containers. So it's a way of kind of segregating things out, and then you can maybe do your experiment on the blue cells and not worry about the green or yellow. So flow cytometry and FACS work like this. And then you get data. Sometimes you get it as histograms, sometimes you get it as these scatter plots. And they're just, I know you can't read this, but it's just basically intensity of marker 1, intensity of marker 2. And that's how they can sort them, and you can take this subpopulation and sort it further and get more and more information. Really what they're doing here is trying to see the percentages of amoeba that have undergone trogocytosis under different conditions. So here's more if you want to look up more on this data analysis. And finally, what their big question is does this, trogocytosis lead to the protection of amoeba from complement? So is this an immune evasion strategy? Please remember, this is complement with an E. Complement with an I is like when I say, great job on your paper. The serum lysis is, remember that in your serum, you have components to the complement system. So they can take a cell, right, and add serum to it. And what they're going to try to show is that when the amoeba takes part of the host cell, it protects it from lysis by complement. And when it doesn't, and when it doesn't have those host cell proteins, it leads to lysis, so it leads to death of the amoeba. So again, big picture is this a method for amoebas to evade the immune system? All right, I hope that gets you through part of the paper, and I hope we have really great discussions. Thanks.